This is where we left off, and we were talking about the differences and similarities, but mostly similarities between mitochondria and chloroplast. And by now, you probably have noticed similarities, and maybe you're getting the two processes confused a little bit, but hopefully we can straighten that out. So mitochondria transfer chemical energy from food to ATP, whereas the chloroplasts transfer light energy to chemical energy of ATP. So again, two sides of the same coin. So that chemical energy of ATP then can actually be used to create sugars, that those sugars then are taken in by heterotrophs, that's us, used by the mitochondria to create ATP. So the ATP that we create, the plants don't get me wrong, but the sugar that we use, the plants made. So the plants get their original energy from the light, they're autotrophs, and they're photoautotrophs. They use that light energy, so the light energy into chemical energy. We take chemical energy of ATP and transfer that into, the chemical energy of food that some autotroph made, and transfer that into ATP. Spatial organization of chemiosmosis differs between the two, but also similarities. In mitochondria, the photons are pumped into the inner membrane space and drives ATP synthesis as they diffuse back. In chloroplasts, the protons are pumped into the thylakoid space and drive ATP synthesis as they diffuse back into the stroma. So all of this becomes kind of important stuff to understand between the differences and similarities between the two organelles. The Calvin cycle itself, I'm not going to ask you each of these products, but I'm going to have you learn something about it as almost an overview, because it's a very important process, but it is something that gets studied in a whole semester by itself, or maybe a portion of a semester. But the idea behind the Calvin cycle, it uses that chemical energy that we made it in the photosystem 1 and 2 of the ATP and NADPH to reduce carbon dioxide to sugar. So that's what the Calvin cycle does. That's its basic purpose. It regenerates its starting material, so that's where it gets the cycle term, after the molecules enter and leave the cycle. So it is cyclical in what it starts with and what it ends with. The cycle builds sugar from smaller molecules using energy in the form of ATP and the reducing power of electrons carried by NADPH, those products that we created already. Carbon enters the cycle as carbon dioxide and leaves as the sugar named glyceraldehyde 3-phosphate. So we call it G3P. Sorry, for short. For the net synthesis of one G3P, the cycle has to take place 3 times, fixing 3 molecules of carbon dioxide. Because you have the sugar name glyceraldehyde 3-phosphate, 3 molecules of carbon. So CO2 has 1 molecule of carbons. We have to turn the cycle 3 times. The cycle itself happens in 3 major phases. You have the carbon fixation phase, you have the reduction phase, and you have the regeneration of CO2 acceptor phase. So the regeneration of the original starting product. And we'll talk about that on the next one. Same image, just have kind of put it on a slightly different way. So 3 phases. The first phase is carbon fixation. The big thing that you put together with that is this chemical called Rubisco. Rubisco. That's actually its nickname. It's a shortened version of its chemical formula. But that has the ability to fix carbon from a low energy inorganic CO2 into a high energy molecule, organic molecule like glucose. It's not quite glucose yet, but a high energy organic molecule. That's what happens in the carbon fixation phase. That's when CO2 goes into the Calvin cycle. That carbon dioxide goes in. Remember, it has to spin 3 times in order to create this final product of the glyceraldehyde. I'm sorry, I already forgot what the name of the final product was in terms of the carbon fixation. I have to go back so I can say that to you. Oh, actually, it's the G3P. Sorry, it's at the bottom. So 3 times. Don't think that goes in and it goes out in the first time. Remember, it has to spin 3 times. But then in that reduction phase, and I'm sorry that I moved the slide for you, in that reduction phase, what's happening, ATP and NADPH provide the chemical energy to the 3 carbon molecules to actually form the bonds. So the chemical energy to the 3 carbon molecules. So you start out with 3 phosphoglycerate. So you have a phosphorus on there, but you have 3 carbon molecules. And then with ATP in, then you have you're adding another phosphorus. So you have a phosphorus on either end. And then what happens with NADH, you then actually remove remove one of the phosphoruses, and then you have a different carbon-based molecule, this G3P, this glyceraldehyde 3-phosphate. And that then is the output of the Calvin cycle. The cycle isn't finished yet, so that's the reduction phase. Basically, what you're doing is you're reducing to reducing ATP and NADPH to their lower state, but you're actually putting energy in to this G3P molecule because it's a higher energy molecule. Then in the third phase, we still have to regenerate into what originally went into the input of the Calvin cycle. So in regeneration is what you do is you're going to take the G3P and with the help of ATP, you're actually going to put phosphates on either end, as well as put 2 of those together to form, again, the Rubisco. So the ribose biphosphate, the Rubisco there, that then is going to start the cycle again. So the important thing, not so much what the individual products are, that you have these 3 phases. First phase is carbon fixation, where that individual carbon from CO2 is going to be put together with 2 others, so you have 3 carbon chain and a phosphate. That then is going to be reduced. You're going to end up with a 3 carbon chain and a phosphate, and also a sugar. That then is going to be regenerated into what then can start the whole reaction again in the Calvin cycle. 3 phases. Now, on from the Calvin cycle into other ways that plants photosynthesize, because certainly you've been asking yourself, and I'm sure you've been saying this to yourself, but I have all kinds of different plants that work different ways, have different sun requirements and also water requirements. These are our adaptions for evolutionary adaptions. Dehydration is very much of a problem because the water is very much of a key component in this in these cycles, particularly the light dependent cycles. There are trade-offs with regard to other metabolic processes, particularly photosynthesis. So if in fact there's the absence of water, you have to ask yourself, what does the plant do? The plant still needs energy, but what does the plant do? On hot, dry days, the plants close their stomata, which conserves water but also limits photosynthesis. So if those pores or those stomata are closed, water is no longer going to evaporate. There is another process that the plant can use. This favors a wasteful, not quite as efficient process called photorespiration. And this one sounds a little bit more like our cell respiration. With this, in terms of photorespiration, that Rubisco, the one that we just talked about, uses oxygen instead of CO2 in the Calvin cycle, producing a 2-carbon compound. So that same cycle, but instead of carbon dioxide being used, oxygen is used, and it's not as efficient, and there is a byproduct that's built up. But most, we call them C3 plants. The initial fixation of carbon dioxide via Rubisco forms a 3-carbon compound. In photorespiration, Rubisco adds oxygen instead of carbon dioxide. So if we look at the images here, the first one is a rice plant, the second one's corn, and the third one's pineapple. The photosynthesis of the C3, you have carbon dioxide moving in and turning the cycle, and the byproducts are starch and sucrose. In C4 plants, the ones in the middle, you have a little bit different design. You have this mesophilic cell. So the C3 has a mesophilic cell also, but this mesophilic cell in C4 has a slightly different compound. It's called a PEP compound that can cause a change in the cycle. We'll pick up that image in a couple of slides. Can cause a change, an adaptation, and that then can turn the cycle. And what happens with that adaptation in terms of the mesophilic cell, that can then cause the waste product to go into the bundle sheath, which can cause the cycle to actually replenish itself. So we have C3, C4, and then we have these CAM photosynthetic plants. Each of them we'll talk a little bit more about. With the CAM photosynthetic plants, you actually have the introduction of the malate, which then makes that particular plant even more water resistant, I guess you might say, or water sparing. So with regard to a little bit more about photorespiration, and we'll come back to those types of plants, consumes oxygen and organic fuel and releases CO2 without producing ATP or sugar. Photorespiration is thought to be a revolutionary relic because Rubisco was first evolved in a time when the atmosphere had far less oxygen and more carbon dioxide. This process limits the damaging products of light reactions that build up in the absence of Calvin cycle. And many plants, in terms of photorespiration, it's a problem. Because on hot, dry days, it can drain as much as 50% of the carbon fixed by the Calvin cycle. So this image next to this information about photorespiration isn't so important that you get it in your head, but I did want to show this relationship, and we will come back to this image on the next teaching time.